Good afternoon, everyone. I'm Rick Barron, the Executive Vice Chancellor for Health Affairs and the Executive Director of the NextGen Precision Health Initiative. I hope all of you had the opportunity to catch our grand opening yesterday of the Roy Blunt NextGen building. What an event. We cut the ribbon, we saw some great performances and videos, and we listened to an inspiring lecture from Dr. Francis Collins, the director of the NIH. Today is our 9th NextGen Discovery Series, and these discussions are intended to highlight critical precision health topics and approaches. We have asked speakers to share their knowledge in ways that engage all of us, and we hope they do so in a manner that sparks new ideas, new collaborations, and new areas of NextGen research. As you hear these wonderful talks, we would like you all to think about research opportunities in precision health where you can contribute and where you can identify potential collaborations across the University of Missouri system. Our featured speaker today is Dr. Brett Froeliger. Dr. Froeliger serves as the director of the NextGen Cognitive Neuroscience Systems core facility here on the Southeast MU campus near the MU reactor and near the baseball stadium. Dr. Choi and I toured this impressive facility last year, and it was a pleasure to spend time with Dr. Froeliger that day and learn about his research. His scholarship focuses on understanding the biologic basis for substance abuse disorders. He is a NextGen Precision Health recruit in neuroscience, and he relocated last year to MU from the Medical University of South Carolina. So Dr. Froeliger is a NextGen naut, as I like to call them, NextGen sailor and discoverer. He has appointments in both the departments of psychiatry in the School of Medicine and the Department of Psychological Sciences in the College of Arts and Sciences. Since 2012, Dr. Froeliger has been continuously funded by the NIH to study brain imaging techniques to evaluate the potential benefit of novel pharmacotherapeutics, behavioral interventions, and non-invasive neural stimulation for treating substance abuse. The NextGen Cognitive Neuroscience facility is a great example of how a lot of what happens in the NextGen Precision Health Initiative is occurring throughout the campus in the UM system, and not only in the Roy Blunt NextGen building. Dr. Froeliger's presence at the MU campus exemplifies that in the area of addiction and that we have talent throughout the entire University of Missouri system, which has numerous federally funded addiction science research faculty. Dr. Froeliger is actively collaborating with addiction researchers at UMSL and UMKC since his arrival. So we asked Dr. Froeliger today to highlight the ongoing efforts across the system to address the challenges of addiction and to also share his own translational research that is focused on new treatments for substance abuse disorders. And with that, I will turn it over to Dr. Froeliger to start today's presentation. Welcome, Brett.
Thank you, Dr. Barron. I really appreciate that introduction. All right, so it's really my pleasure and privilege to be able to be here today and talk with all of you. That was a fantastic introduction, and I really also enjoyed the festivities yesterday, and in particular, getting to hear Dr. Collins talk to us about the future of precision health. So let's get some housekeeping out of the way first. Some disclosures and funding. I'm required to say I do not receive any money from the tobacco industry. I'm primarily a tobacco researcher. That is the cornerstone of my research, and I do not receive money from the industry. I have received consultant services from Promentis Pharmaceuticals within the past 2 years, but that work is unrelated to the work I'll be talking about today. And then all my research has been funded by the National Institutes of Drug Abuse, NCCIH, and NCI. So we all know and have heard that we're facing a public crisis, health crisis. And some of that is focused on the opioid crisis that we've seen throughout the state, throughout the country. And the nature of the problem has shifted over the course of the past really 15 years, from prescription opioids to increased usage of heroin to now synthetics. And so what has maintained and what continues to escalate is the fact that people are addicted to opioids and people are overdosing due to opioids. And the factors associated with that are quite complex, and I will not get into that today, but we have experts from across the state that are focused on this problem of health disparities, underserved populations, and how do we get treatment to more individuals who are vulnerable to overdose and poisoning deaths from opioids. But the other crisis that I really want to focus on as well is what I would call our cancer epidemic that's associated with tobacco use. And so I will be focusing largely on cancer-related, or I should say tobacco-related impacts on health, and some of the work that we've been doing to better understand why is it that a person continues to smoke despite the fact that they may want to quit. Well, as I said, that there's a crisis. The crisis truly is in our front yard. I came here, as Dr. Barron said, last summer. I arrived here on the ground. It was mid-June. But when I was considering the possibility of leaving my former institution, I looked around to see where is it in the United States is there the greatest need. And unfortunately, Missouri is one of 7 states throughout the country that has the highest prevalence of opioid deaths, but also smoking. And there's, again, a number of factors from policy to culture to a history that have been contributing to that. So there's a large need right in our front yard that we need to address. So for all of you who were able to attend the NextGen opening yesterday and hear Dr. Collins talk, the NIH has been funded by Congress, approved funding, which has been increasing over the course of his tenure at the NIH. But unfortunately, there's other needs besides just drug addiction. And the NIH, and NIDA in particular, has been a primary funder of addiction research in the country. And with these competing needs, you can see in the graph to the left that 2021, the funding line for NIDA, in fact, decreased. And if you look to the right, you can see, while it may not be a large percent decrease, research project grants and research centers, the funding for those mechanisms have also gone down a little bit. And so looking at this, this hopefully will change as a function of support from Congress, but also when we address some of our other critical needs that we're currently dealing with. But in the meantime, we need to be able to become smarter and more effective of pooling together resources and expertise to be able to make the most out of the dollars that we are given. So one way to accomplish that, and I'll thank Dr. Barron for initiating this effort, is developing our addiction science research interest group. And Dr. Barron put us in contact with Mary Hindle, and she's been pivotal in helping us get together a group of investigators from across the system that are all focused on substance use disorders, treatment, and dissemination. And so this group is led by myself, Dr. Dennis McCarthy, and Mary Hindle here at MU, Dr. Rachel Winograd at UMSL, Dr. Holly Hagel at UMKC. And we've had over 70 people that are now a part of this interest group. And roughly half of those 70 people are attending each monthly meeting, depending upon the content. But what's really fascinating and enjoyable for me to see is that this spans over 16 different areas or disciplines. And so this isn't focused just in psychology or sociology or pharmacy or schools of medicine. This is truly a group effort that we're coming together and talking about how is it that we can pool our resources and our expertise to make a bigger impact in the state of Missouri and nationally. So first of all, I'd like to highlight Dr. Dennis McCarthy and his leadership for MOCARE. This is the Missouri Center for Addiction Research and Engagement. This is a university-funded center, and the MOCARE was developed last year. There's It's housed in the Department of Psychology, and there's a 30-year history of addiction research and training at MU. There's 10 faculty that are focused on addictions, from behavioral genetics to mobile technology to quantitative methods. And in fact, I need to highlight Dr. Kenneth Scher, who is really one of the seminal researchers in addiction in the world. And he has been here and been a pioneer of addiction research, and we get to leverage his expertise in continuing the development of our program of research and treatment development. Among the group here at MU, there's multiple NIH R01 funded investigators. There's a prior NIAAA P60 Center grant, and there's a new P60 Center grant that is in preparation for submission this fall that's being led by Drs. McCarthy and Scher. There's a T32 here that's NIAAA funded, and what that does is that provides support for trainees from pre-docs to postdocs, and allows these individuals, these students, our mentees, we can develop their expertise and give them training time and learn about becoming the next generation of addiction scientists. And importantly, there's also an R25 summer program that's been ongoing for 10 years. And so there's a great breadth of expertise in research and also in training that's found here at MU. But it doesn't stop here at MU. At UMSL, Dr. Rachel Winograd is a part of a number, or in leads efforts on a number of different programs. And her programs are looking at ways to better reach individuals that are struggling with opioids and are vulnerable to opioid deaths. In fact, one project, the MoHope project, involves overdose education and naloxone distribution. And she had recently reported to our group that they've gathered reports and working with the state that they have contributed to the reversal of over 8,000 overdoses. That is truly fantastic, and the impact that's having on public health is phenomenal. And last, I'd like to highlight the work that's coming out of UMKC. And our leader on our interest group is Dr. Holly Hagel, and she works with a larger team, and they're a part of the collaborative, as they call it for short. And it's to advance health services. And the collaborative is a network coordinator office for transfer content. So they support dissemination and education and getting treatments out to the public. They have 23 federally funded projects totaling funding of over $40 million. And so we have this network in place throughout our state, throughout the UM system, to really make a fundamental change and difference in the lives of Missourians, also for the lives of those that can be used as a model to help improve the health and well-being of those suffering from addiction throughout the country. So as I was talking about some of the work that's being done, we know that there's a number of evidence-based first-line treatments for opioid use disorder. And getting these treatments out to the public is essential to save lives. That work that's being done by Dr. Winograd at UMSL is an example of that. For tobacco use disorder, we also have a number of first-line treatments that have been FDA approved that are quite effective. And what we see is that if we can get these effective treatments to smokers and to individuals that are exposed to opioids, we can actually help save lives. We can help them reduce their use and hopefully prevent a relapse once they become abstinent. Unfortunately, however, the majority of people, once they've developed a significant substance use disorder, relapse even after receiving a first-line pharmacotherapy. So one of the questions I ask myself is, what can I do? What can we do to better understand new treatments, the etiology of addiction, and how can we better treat the pathophysiology of substance use disorders to help augment and further advance the treatments that are available that can be given in the examples that I've already demonstrated. So I will pivot slightly to talk about my research. My research, I have a goal, which is I want to understand and examine the neural mechanisms of action, or MOAs, of promising new therapeutics for addiction. We're not trying to recreate the wheel. Rather, what we're trying to do is, if we can better understand the basic fundamental brain mechanisms and neurobiology underlying the ongoing use of addiction, understand how it is that new treatments may modify those neurobiological mechanisms, then we can use new treatments or adjunctive treatments to increase the clinical efficacy of treating drug addiction. So we overlay brain imaging onto brief randomized controlled trials. So for many of you who've been engaged in clinical trials, it's known that it takes a significant number of years to develop and test whether or not a treatment, a medication, an approach may have clinical efficacy. It's expensive, it requires a large number of people. So I focus on these brief experimental designs to where we can screen medications and approaches quickly with a smaller number of people, which I'll explain in a little bit. And if there appears to be a modification of the mechanism on which we're trying to treat, then that can initiate a larger scale clinical trial to test for clinical efficacy. So our outcomes are brain mechanisms underlying treatment effects and drug use behavior, and the behavioral outcomes as well. So as I mentioned before, the canonical translational design is if you develop a new medication, for example, and you evaluate it preclinically, determine safety, usually in preclinical in phase 1. And in phase 2, you begin to test in a small number of people whether the mechanisms of action that support the indication are indeed found. After which you can move on to phase 3 trials. So you can see where I position my lab. And the idea for doing this in part is because then we can take those findings and we are the middle person. My laboratory does the work that we can then back translate, say, into a rodent model to better understand the neurobiology that underlies this neural circuitry changes that we're seeing in the brain. But we can also forward translate that into larger scale clinical trials, working with clinicians at MU, throughout the state, or throughout the country. I have to pivot again and answer this question, why MU? As I had said before, as Dr. Barron had indicated, I moved here last year from a productive location. Well, there's a need. Just in the past year, and this is pandemic numbers, is that we pre-screened 947 nicotine-dependent adult smokers. And that's just within the Columbia and outlying Columbia area. So while there's a long history of addiction research here at MU and throughout the state, a primary focus has been on alcohol use disorder or alcohol use disorder susceptibility or opioid use disorder. And so as far as smoking research, we have a ripe opportunity to reach people. Then, of course, the expertise in addiction science. As I had already indicated, this expertise extends across the system. Leadership. The NextGen Precision Health and the neuroscience focus is extraordinarily attractive. It was extraordinarily attractive to me, but also it's not just the building. The building is a fantastic building, and the resources that are going to be in there are fantastic as well. But it's really the people that are supporting it and the people that are going to be doing the work in there, which was highly attractive to me. An institutional commitment. From the top all the way through really from the top down, I've had a uniform message of a commitment to addiction research through MOCARE, the support of the new Cognitive Neuroscience Systems core facility, which I'll talk about more in a moment. And that was also very appealing. And then last but not least, Columbia is a great place to live. This is a place that the work we do is time consuming and it's hard. And when we're not doing that work, we need to be in a place where we can enjoy the people that we love. So the work that I'm doing is all conducted within the Cognitive Neuroscience core facility. And so what this is, there's a research dedicated Siemens 3T Prisma scanner. There's a MOC scanner, transcranial magnetic stimulation suite, and I'll describe that more in a little bit, or TMS. We also have a brain site neuronavigation system to where we can scan people. We can identify areas in the brain on each individual subject of where we might want to stimulate using TMS. There's a 256 channel EEG system, a full suite of psychophysiological recording equipment, multiple experimental testing rooms, clinical rooms for interviews and biospecimen collection, and then an area to store those biospecimens. Now, what makes this unique and what was very attractive to me is that there's a number of institutions around this country that have these components. But having all of this in contiguous space allows for truly translational work between basic science to clinical applied translational science. Now we're new. We opened last year, and right now there's 7 different active studies that are currently ongoing in the CNS. But the exciting thing is we're growing. So there's 7 pending NIH grants that are looking to rely on the research being conducted here at the CNS. So just some quick examples of what is it that MRI can do in the context of understanding substance abuse and treatment outcomes. Well, of course, we can look at brain structure. So this is not unlike what you would do if you went in to get a scan at the hospital if you had an injury. So we can look at brain structure as a predictor of treatment outcomes, or potentially as an outcome from chronic exposure to drugs of abuse. We can also look at functional MRI. So this is where we tune the scanners differently to be able to be sensitive to blood flow in the brain. And we can examine changes in blood flow as a response to, say, a stimulus that a person would see while they lie inside the scanner on a mini TV screen. But then we can also look to see how it is that areas of the brain that become activated through the functional MRI, how those areas of the brain that we know are part of neuroanatomical circuits are functionally communicating or functionally connected over the course of resting periods or during periods when they're trying to process information. So I apply that approach and those techniques to understanding what I call to be transdiagnostic mechanisms involved in drug abuse and addiction treatment outcomes. This is reported from, or this slide comes from the National Institutes of Drug Abuse. And so while this little cartoon is overly simplified, it does get at some of the core features that we need to be focusing on when we're looking at the neurobiology of addiction. Today, I'll be focusing on inhibitory control and be talking about work that I do with fMRI, TMS, and looking at addictive behavior. And also reward, where we use fMRI and a behavioral medicine approach to look at addictive behavior outcomes. So let's talk about inhibitory control. This is, as it sounds, it's the ability to inhibit and control your behavior, or to suppress maybe an overlearned motor output, a thought, an action, or a feeling. This particular example that I'm giving you here is a way that we can probe this inside of the scanner, and others do this across the country in fMRI or EEG or other laboratory environments. You ask a person to press a button rapidly every time they see a gray circle, which happen very frequently and very quickly, or repeatedly. So every 400 milliseconds, they're seeing the circle, they're pressing a button. But then periodically, they have to inhibit their button press when they see a no-go or a blue circle. There's been about 15 years of research that have indicated that there's a hyperdirect pathway in the brain that comprised of the right inferior frontal gyrus, as you can see to the figure to the right, the pre-supplementary motor area, the thalamus and subthalamic nucleus, that these areas of the brain are structurally connected between nodes, between the right IFG, the pre-SMA to the subthalamic nucleus, and the subthalamic nucleus to the thalamus. But they're also, in addition to being structurally connected, functionally communicating to subserve successful inhibition of behavior. So if we use this hyperdirect pathway as a model by which we can examine how is it that an individual with a substance use disorder, and substance use disorders broadly, have been shown to individuals have been shown to exhibit deficits in inhibitory control, or varying levels of deficits in inhibitory control, we can begin to ask questions about, are there individual differences in the circuitry function that may be associated with the prevalence of smoking, opioid use, or relapse vulnerability? So earlier in my career, I conducted a number of studies among individuals that were long-term heavy smokers. We had them perform a task like this and other tasks that probed minor alterations of the process of inhibitory control. And we found that compared to non-smoking controls, the individuals with a tobacco use disorder exhibited hyperactivity in the inferior frontal gyrus of the brain. Now, this is interesting because in the basic, or in the non-substance abusing literature, we've seen that activity in the inferior frontal gyrus, as it increases, that's associated with better inhibition. However, among our smokers, we're seeing that hyperactivity in this area of the brain is compared to non-smokers is in fact associated with worse performance. And others have found this as well. Groups out of UCLA have reported these findings back in the around 2015. And the idea is, without expanding upon the interpretation, is that perhaps it's a compensatory mechanism, individuals trying harder to exert inhibition. In 2016, Rita Goldstein's group out of Mount Sinai evaluated the literature among individuals with a substance use disorder who performed some form of an inhibitory control task, and found that consistently across different drugs of abuse, individuals exhibited this hyperactivity in the inferior frontal gyrus and the pre-SMA, multiple nodes within this hyperdirect pathway, that those who did actually were those who went on to relapse following a quit attempt. So there was a slight indication, this was all retrospective analysis, but there was an indication or an idea that was developing that perhaps what's happening here is this may be a biomarker for relapse vulnerability. So what I chose to do is follow up on this line of work and ask the basic question of what is the role of inhibitory control neuroactivity in the hyperdirect pathway in smoking relapse vulnerability. So we asked this question and executed it in 2 studies. The first was a smoking cessation trial, which recruited 81 people that were interested in quitting smoking. Everybody came into the laboratory, they performed the task that I explained to you inside the scanner, and 30 days later, they made a quit attempt. After they made the quit attempt, we tracked them for 10 weeks after their quit attempt, and we evaluated who is it that remained abstinent and who is it that relapsed to smoking as indicated by 7 days of continuous smoking. In the second study that we did is that we performed a laboratory-based relapse or lapse analog study. Here, all the participants came in, there was 26. They went into the scanner, performed the task, but rather than 30 days, we waited 30 minutes. And 30 minutes later, we provided them with a bunch of cues that were associated with their smoking, such as their preferred brand of cigarettes. We bought them, we packed them, we pulled one out, we gave them an ashtray and a lighter to essentially tempt them to smoke, but we paid them not to smoke. And so what we did is we tracked how long would a person go and remain abstinent and receive money for remaining abstinent versus saying, I'm no longer going to earn money over the course of an hour, I just want to smoke my cigarette. And we evaluated the brain mechanisms during inhibitory control in each of these studies as indicators of how long a person would go before they lapsed in study 2 or full-blown relapse in study 1. So just briefly, I'll show you the demographics. For those of you who may be familiar with smoking research, I'll just indicate real quick that the nicotine dependence is measured by FT&D. Everybody was moderately nicotine dependent and smoking on average between 15 and 20 years, and roughly 3/4 to a pack a day. So the first question that we saw found an answer for is that this hyperactivity in the right inferior frontal gyrus and the thalamus, 2 nodes of the hyperdirect pathway, the hyperactivity was associated with those who went on to relapse. Those who remained abstinent had relative less activity in this network while performing this task. And then in study 2, the laboratory-based study, the hyperactivity was also associated with smoking soon in the laboratory, only 30 minutes after they performed the task. So in other words, this hyperactivity appeared to be a risk factor for lapsing and full-blown relapse. But the hyperactivity still did not answer the question. So in some regard, this recapitulated or just repeated what had already been found in the literature. So we're really interested in how is it that these 2 areas may be functionally connected or communicating over the course of performing the task. Is the circuit somehow dysfunctional? And is dysfunction within the circuit explaining the variance in relapse vulnerability? So then we conducted an analysis to look at the connectivity, the functional connectivity between these 2 areas of the brain. And we found that those that remained abstinent had stronger functional connectivity in this hyperdirect pathway as compared to those that relapsed. But in the laboratory-based study, we replicated that, showing that stronger functional connectivity in this pathway in the context of inhibitory control also was associated with going longer without smoking a cigarette. And then when we looked at that, the kind of the triangulation of those findings, what we found is inhibitory control performance on the task was associated with the smoking outcomes in both studies. But what explained that association of inhibitory control performance and smoking lapse vulnerability was the strength of functional connectivity in the brain. So what that told us is that we have a circuit. We have a circuit, a brain circuit that is explaining significant variance in behavioral inhibition and relapse vulnerability. Then the next question is, well, what can we do to strengthen the circuit? And if we strengthen that circuit, does it reduce relapse vulnerability? So transcranial magnetic stimulation is an FDA-approved device for treating major depressive disorder, OCD, trichotillomania, and most recently, as of August of 2020, a form of this has also been approved for treating tobacco use disorder, short term. Now, TMS, as it's been FDA cleared, the protocol is roughly 20 minutes. So it's a 20-minute session. A person comes into the clinic, they get TMS for 20 minutes, and this could be repeated multiple times a week over weeks in order to reap a clinically beneficial outcome. But in 2005, there was a paper that came out in Neuron that showed that if you, in fact, rather than just doing high frequency or low frequency TMS, which is done in the clinic, if you take a Theta Burst stimulation, this is a pattern TMS frequency, you can reduce the duration of the stimulation. For an excitatory pattern, it's roughly 3 minutes. For an inhibitory-like pattern, it's about 33 seconds. And each individual stimulation, the 33-second or the 3-minute stimulation period, it's been shown that those effects, one stimulation, the effects last around 1 hour. So the acute effect is only 1 hour. However, what this means is that if you can reduce the clinical intervention from 20 minutes down to either 30 seconds or 3 minutes, that's a significant benefit not only to the patient and the healthcare providers, but also our healthcare system. So what we decided to do is use more of a precision medicine approach. We recruited people, smokers, to come into the laboratory. They could get an fMRI while they perform the task I explained. And then what we do is that we can individually identify within a person's brain the peak maximal activity that's involved in performing the task. So as you can see, there's a lot of overlap in the area that's in the crosshairs between these three example participants. But there's so much variation in brain anatomy, the folding, the location, even the functional location of performance of some of these tasks. So now we can get greater precision of where to stimulate using TMS. Here's an example of myself in the chair and Alex Brown, who works in the CNS core. And we can load these data that I'm showing you from figure 2 onto our screen, and now we can use neuronavigated TMS for precision. And in doing this, in a study that I'll just discuss with you momentarily, in 37 participants, we can track how close we are, or the target error distance, from the functional activity as shown in figure 2, and we can get within 3 millimeters consistently. Then, once a person is completed with their acute TMS treatment, we can put them back into the MRI scanner, have them perform the task again, and examine the effects of TMS on brain function. And then finally, we can assess clinical behavior. And there's many different ways that we do this, but outside of the laboratory, we can track individuals' behavior. We can look at side effects, smoking, and craving behavior outside of the lab. So in a study that we recently conducted here, this was a NIDA-funded UG3 study. We recruited people into the laboratory during the screening assessment period, doing extensive background health screening. We provide them with training on the procedures. We have them perform a baseline MRI scan where we analyze their data, and then those individual subject data are used to then guide the neuronavigation TMS. And then everybody came in on 2 additional days to receive either the excitatory, which is ITBS, or an inhibitory, CTBS, form of Theta Burst stimulation. So they were scanned on each experimental visit. They were given the TMS and then scanned again to look at the acute effects of treatment. So I've never before reported results of a participant enrollment table, but I've never before transferred a program of research in the heart of a pandemic. So in fact, this is one of the slides that I'm most proud of, and I'll explain the reason why. So the study began in the Southeast. And when COVID hit, we shut down, and I believe the last day was March 8th. So March 8th, we were done recruiting people. But shortly before that, I had already been in conversation to come here to MU, sign agreements. So I was preparing to transition the project, but COVID hit, everything shut down. So we have no enrollment. And in fact, what you can see from the black line, the planned enrollment, we were at risk of the study failing. It was late June, early July before we had boots on the ground here. I think I pulled up in a vehicle and the latter part of June, a hired team. We did all this via Zoom. We all got here, and really thanks very much to administrators, construction services, working together. The CNS was open for business in mid-August. So looking at this, none of this would have been possible if it wasn't for the fact that we had a huge team of people working together, both within my lab, but really across departments, colleges, and MU to really help make this happen. So first of all, the results of the safety profile, it's safe. If you look at the black line, that's the baseline visit. These are self-reported symptoms following the pre-TMS. So this is when people came in to get their brain scanned for us to be able to take those data and characterize where to subsequently stimulate. We assessed at the beginning of the visit, the end of the visit, 24 hours later, and 48 hours later. And you can see that people actually report higher symptoms when there was no TMS. When we did the CTBS and ITBS, there was no significant increases. Tolerability is well tolerated. Over 90% of the people return for each visit. And as I was talking before about how we demonstrated that it's this hyperactivity in the inferior frontal gyrus that is associated with relapse vulnerability. When we use an inhibitory form of TMS to the inferior frontal gyrus, so we target that brain region, what we see is that it improved smokers' inhibitory control. But when we use an excitatory pattern, the ITBS, it in fact made their behavioral inhibition task performance worse. So if we move that forward and now look at the effects of the inhibitory form of TMS, Theta Burst stimulation, on their naturalistic smoking 24 hours after they leave, we saw that it significantly decreased their smoking behavior as self-report after they left as compared to their baseline visit. Similarly, 24 hours after they received the TMS and left, and we assessed their craving to smoke, it significantly reduced their craving. CTBS also strengthened the functional connectivity in this hyperdirect pathway between 3 of the nodes. And the extent to which it did was associated with these behavioral findings that we have with regard to the reduction in smoking and the reduction in craving to smoke. Now, I'll be the first to admit, these findings were somewhat surprising to me because we're only looking at a 33-second long stimulation parameter, which we anticipated would only last for roughly 1 hour. And we're seeing an emerging effect that's extending beyond after a person leaves. So those data formed a subsequent R01 application that we're looking at to be able to now evaluate the clinical course of TMS for helping people quit smoking. Now, there's a lot going on the slide, and I apologize, but I wanted just to point out a couple things, which is rather than the 1 session, we'll have 12 sessions over the course of 3 weeks. The treatment duration, as is what I said, and people may receive treatment up to 5 times a week. After they go through this three week period of treatment, they will have a quit date set. And we'll look at post-treatment, the effects on, we'll biochemically verify their smoking via carbon monoxide, breath carbon monoxide levels, saliva, so we can look at the nicotine metabolite cotinine. We'll have questionnaires. We'll examine the effects on the brain by having them come in for a visit. But then we'll also do a clinical follow-up out to 6 months. And so this allows us to be able to take these basic mechanistic findings of the effects of TMS on brain circuitry and relevant behavior, and now test a clinically therapeutic course of TMS to see if it improves smoking cessation outcomes. Now I indicate here Aspire data collection, and I want to talk about that briefly, because while I focus on brain mechanisms that explain behavior and treatment outcomes, in 2021, and as everyone knows here, the world has changed. It's changed with regard to the technologies that are available. It's also changed with regard to how is it that we can actually assess people, and how is it that, or what it is that people are willing to do. So we've been working with a colleague of mine, Dr. Valafar at USC in Columbia, South Carolina. He's a computer scientist. We published a paper recently showing that if we give a person a smartwatch and just utilize a bunch of the sensors on the smartwatch, we can characterize whether a person is smoking a cigarette as compared to chewing on a straw or eating food or drinking water. We've tested this algorithm uses neural networks, machine learning to be able to learn the behavioral gestures associated with smoking. And so we validated this technique to where now we can passively detect whether a person's smoking, how much they're smoking, how frequently, the number of puffs they're taking, where they're smoking, and then also when they're smoking. So now we can develop not only characterize their smoking topography in the natural environment, but now we can begin to build prediction models as well. I'd like to recognize the team of researchers that are helping with this project, and this is across multiple departments and colleges. And I would like to highlight one of our newest faculty members to join MU, Dr. Melissa Terpstra. And she's recent to this team, and she brings a great deal of expertise in MR spectroscopy. And so one additional components of this work moving forward is to look at the effects of TMS on GABA and glutamate in the brain, and her expertise in that area will be invaluable. So now I'm going to pivot to the second point that I want to talk about today with regard to my research, which is reward processes. So it's long been known, and there's a seminal paper, a science paper in 1997 by Wolfram Schultz. If you look at the left, the upper left box, you'll see that these dopamine neurons in the ventral tegmental area, this is the area of the brain that is dopamine neuron rich, that when, this is not humans, that when presented with a rewarding cue, what happens is those dopamine neurons begin to fire. However, if there's a stimulus that's paired with that rewarding cue, a conditioned stimulus, such as a light, a picture, something that's consistently paired with that rewarding cue, if you go down to the bottom figure, you'll see that the reward doesn't necessarily elicit a response in those dopamine neurons, but the stimulus that predicts the reward does. So what this means is, this is a fundamental premise in the addiction literature, is that drugs, which is a reward, can produce a reward response. But the cues that are associated with the availability of drugs can begin to elicit a response independent of the actual receipt of the reward of the drug. Translating this to humans, in an older meta-analysis of fMRI data, it was shown that in the ventral striatum, a dopamine-rich area of the reward circuit, that individuals, when presented with a variety of unconditioned to fully conditioned cues, consistently across the literature exhibit increased fMRI bold response in the ventral striatum, similar to what was shown by Schultz and colleagues in Science. Now transitioning to smokers. So Elliot Stein's group from NIDA Intramural published a paper in 2012 in Biological Psychiatry showing that if you give smokers and non-smokers juice inside of the scanner, and you look at the bold response in the ventral striatum, the nucleus accumbens is a region within the ventral striatum that we believe is a seat of reward processes or responsivity. That compared to the non-smoker controls, smokers exhibit a blunted activity in the nucleus accumbens to reward cues, to non-drug reward cues. And in fact, the cues that predict the non-drug reward, in this case juice, also fail to elicit a response in the nucleus accumbens. Unfortunately, we also know from the literature in a meta-analysis that nicotine, alcohol, cocaine, and this is extended into opioids and methamphetamines and other drugs of abuse as well, is that for those individuals who are addicted to that particular class of substance, if you show them a cue that's associated with the availability of their drug, a pack of cigarettes, a bottle of alcohol, perhaps a needle or pills or a patch, fentanyl patch, that will elicit a response in the nucleus accumbens as you depicted up above in the top half of the slide. So that brought my colleagues and I to ask this fundamental question, well, what could we potentially do to be able to reorganize or restructure an individual's response to the type of reward and what they find rewarding and the items in their environment that may predict reward. And so behavioral platforms have been shown to be terribly successful or incredibly successful in helping people to restructure and reappraise and reconfigure their interpretation of their association with things in the world and their thoughts and feelings. But one area that I have found or had found at the time to be woefully underrepresented is teaching people how to reappraise the natural value of non-drug reward among people who are addicted to drugs of abuse. In savoring is a principle that I was attracted to. I found an individual that was developing a manualized treatment to focus on savoring natural reward in place of focused attention on drug-related reward. And so the basic idea is you're using a metacognitive attentional control process where you use your prefrontal cortex to reorganize thought patterns around what is rewarding, what are your goals, and really try to amplify and focus in on savoring non-drug related reward. So this treatment that I briefly mentioned is called mindfulness-oriented recovery enhancement, or MORE. It involves mindfulness to regain attentional control, reappraisal, so increasing psychological flexibility, and then savoring of natural reward. It involves mindfulness, CBT, and positive psychology. Now, since this was developed and I began working with Dr. Garland, there's 26 papers published on the effects of MORE in substance use disorders, with the majority of those being in chronic pain patients who developed prescription opioid misuse or opioid use disorder. And again, I just highlight for those of you interested in behavioral treatment platforms that MORE, not unlike CBT, focuses on a number of processes such as mindfulness of stress or attentional awareness of automatic habits. There's the mindful reappraisal. The savoring is one of the key components in this particular treatment that I'm interested in. And these are executed in group sessions that are about an hour and a half long, led by licensed therapists. And then people are given homework of 15 minutes of mindfulness reappraisal, savoring practices a day, encouraged to do so. And in the context of our smoking studies, we ask people, before you smoke a cigarette, please use 3 minutes of mindful breathing before you do so, or 3 minutes of your mindful savoring practice before you do so. So first of all, I'll show you some findings from our study looking at the restructuring reward in opioid misuse with a MORE treatment. So this is a recent completion of a number of studies that were funded by the DoD and NIH among individuals with chronic pain that were using prescription opioids who had opioid or reached criteria for opioid misuse, compared to a social support group. The group receiving MORE had a significant reduction in their brain response as measured by EEG, or electrical cortical activity in the brain, in response to drug cues, opioid cues in particular. And they also had a significant reduction in their self-reported craving following treatment. With regard to positive emotion brain response, we see that compared to the support group, the MORE group had a significant increase in their brain response as measured by electrical cortical activity using EEG to those cues compared to the support group. And they also reported higher self-reported positive affect. Now, we found that as compared to social support, MORE led to a reduction in opioid use that persisted 3 months post end of treatment. But what was fascinating is that the treatment effects on the reduction in opioid use were statistically explained or mediated by the changes in the brain response to positive emotional cues. So this provides us an insight into if we treat or help restructure reward processes among individuals with a substance use disorder, in particular opioid use disorder, that can be a therapeutically relevant approach to augment other treatments. Similarly, we did a study with smokers, and we found that compared to our control group, the individuals that received MORE had a significant reduction in their brain response to smoking cues following treatment. Now, these smoking cues are typically very potent, and so this was very exciting for us. But in the same areas of the brain, we see that the smokers who received MORE had a significant increase in their brain response in the reward circuit to positive emotional images. And those changes in the reward circuitry of the brain, from going from having a more blunted response to drug cues to a more elevated response to positive emotional cues, explained some of the variance, or was associated with, the increases in positive mood, the reduction in urge to smoke, and in fact, a reduction in smoking behavior prior to making a quit attempt. Now, what we're doing now is following up on that and having a more rigorous control group. So we're currently underway recruiting smokers into the laboratory here at MU. And this is being led by Stephen Craig, who works in my lab. We're recruiting smokers from the community to come in, and they receive semi-weekly group meetings, either for MORE or they're getting cognitive behavioral therapy. And we scan them at the beginning and at the end, and then we assess outcomes. So I hope next time I'm able to speak with any of you, I'll be able to share findings of that study with you now, or at that time. Now, I'm mindful of the fact that we do live in a drive-through society, right? Having 8 weeks, 4 weeks, 8 weeks, 10 weeks of treatment, of behavioral treatment that's intensive, may not be amenable to all people. So as I indicated, I was interested in savoring. So we're focusing on savoring. We're seeing, can we use savoring, this capacity to attend to joys, pleasures, and other positive feelings? Can we use that and brief savoring as a mechanism or as an approach to help people recover from substance use disorders? Here's an example of this. I won't go through this now, but for those of you who have access to this slideshow later, this is also posted on the NIH HEAL initiative. It'll walk you through how to savor. What's fascinating is before this ever even approached or came into the discussion in the context of addiction, Mauricio Delgado's group out of Rutgers reported in a Neuron paper a number of years ago that when they taught people that didn't have a substance use disorder how to savor and asked them to savor in the scanner, in the MRI scanner, what they found is yes, people reported feeling better when they were savoring, but that was driven by an increased response, again, in the ventral striatum brain area, ventral striatum reward area of the brain. In Dr. Garland's clinical trial that I'm a co-investigator on that I was talking about, where MORE is being used to help treat pain patients with opioid misuse, a significant number of people were also smokers. So we looked to see, does MORE compared to social support reduce smoking behavior among those people, even though that wasn't a concrete or talked about behavioral directive. And in fact, it does. So then we followed up on that. We conducted a small randomized study where We wanted to see if we give brief saver training to smokers in the rural community. We did this in South Carolina over the past year. We had a licensed clinical psychologist contact everybody and recorded a saver training, kind of a 30-second clip of how to saver, talked to them, the control group. We provided them with skills and techniques for quitting smoking, kind of standard of care. Everybody in the community that we reached out to, these 44 people, we also mailed them nicotine replacement patch as a platform pharmacotherapy treatment. And then asked the question, if given freely to people, nicotine patch, by just adding this brief saver training, will this improve or change smoking behavior in our rural population? And what we see is that compared to the control group that was on and received a nicotine patch, the saver group made more quit attempts over the 4-week period in which we assessed them. And they also reported a reduction in craving to smoke after the 4-week period. We'll continue to do this work both in our R01 clinical trial, looking at more and looking at the dose effects on savoring. We're continuing with this work. We'll be looking at brain imaging to better understand among smokers, how is it that savoring may augment reward processes, and how may that translate to treatment outcomes. And we're also in the process of a multi-site team science center grant to NCI, which will look at the brain mechanisms underlying savoring effects here in Missouri. And that's project 2. But in project 1, this will be a multi-site trial where we're sending out, similar to the study I presented to you earlier, we'll be sending out nicotine replacement therapy to rural smokers in South Carolina and in Missouri, and we'll be providing them with a behavioral platform of brief saver training or just informational packets of tips and tricks of how to quit smoking. As far as this PO1 team science, I want to thank Dr. Kevin Everett, who has been a great colleague here at MU, who is heavily invested in helping smokers to quit smoking. And he has joined the team as a co-investigator. I'd like to acknowledge the Ellis Fischel Cancer Center for advisory support and program development, and Dennis McCarthy in MOCARE for program development as well. Now I'm nearly done, but I'd be remiss to not speak to this issue of reproducibility and translation. We have this great breadth of expertise throughout the state of Missouri that's working at on each of our campuses. But we have this problem, and NIH has brought this to, I think, everyone's attention of all these findings coming in that are lacking in reproducibility. How do we translate these basic findings from preclinical to laboratory, human, basic, to clinical and applied outcomes? Well, I'm not convinced that this particular graph I'm showing you is the answer, but it is a great framework that's put out by the National Institutes of Mental Health. It's an RDoC framework for those of you who have not seen this before. Part of the issue I think that we're having is that we have our ways of going about asking our questions, the techniques that we're using, the boundaries of how we operationalize the questions, and the analytical strategies that we take to try to answer those questions. The RDoC framework is a framework that lays out, here are levels of analysis that we can focus on, and here are domains by which we can focus examining and investigating those levels of analysis. Dr. Collins' talk yesterday talked about these large-scale NIH initiatives, which will be getting large amounts of data from a large number of people, and we can access that. To the extent to which we're looking at genetics, we're looking at molecules, cellular function preclinically, we're looking at neural circuits in humans, physiology, behavior, self-reports. If we communicate and if we collaborate and come up with some standard guidelines of what we would like to do and participate to collect like data, to harmonize to the extent to which we can up front, we can pool those data, not just in addiction sciences, but across different areas and domains to better understand human health and behavior. So I get the pleasure of talking with all of you and giving a brief survey of the addiction work that's going on across the system, and and highlight just two of the projects we're doing in my lab. But the fact is that without my research team, I would only have ideas to share with you and nothing concrete. So while you don't get to see them, please trust me when I say I see them truly outstanding in the field of tobacco. Lastly, I want to acknowledge support from MU. As I said before, I came here in the midst of the pandemic. It was a crazy time for everybody. And so I want to recognize the support from President Choi, from some outstanding chairs, Dr. Arndt and Young Walker, the support from Deans Drury and Zweig, Dr. McIntosh, helping bring together the equipment and infrastructure into the core facility, Dr. Barron for really spearheading and helping us and get together this addiction research group and bring parties together. And then Dr. McCarthy, the director of MOCARE, for really being the glue of addiction science. I feel that's he's the one who's known everybody. He was doing this work long before I ever came here, and some of us were ever in parties here. And then lastly, I want to acknowledge Mike Sternello. He was the person that when he was given the green light to get the core facility and the lab outfitted and work through all the details and construction, he figured out a way to get it done. And it was a really complicated time. So I want to appreciate him and everybody else that's provided support. Lastly, if you want to learn more about addiction science, please contact myself, Mary, or Dennis. We'd love to include you as part of the conversation. And thank you very much.
Thanks so much, Brett. That was a great look at what you're doing here and across the system with partners. The addiction group under your leadership and Dennis's is our poster child for how we want scientists to collaborate across the system. So I couldn't be more thrilled at how you guys are working together. The amazing recruitment you had after you opened your doors back up, how did you advertise?
Yeah, so what we did is advertised everywhere. So we did TV, we did the election. We had a lot of viewership during the election. So we did the election, we were doing NFL games. So a lot of TV, we did Craigslist, Facebook, we do posters at the mall. Any place where we can get advertisement out there, we do it in every format we possibly can.
Well, we need to get a list, Mary, of all the ways he advertised. And for, although we don't want to take all your secrets away, but it is an art to figure out how to recruit people through those communication vehicles for studies like this. So you guys clearly killed it. Do you think you're successful because of those techniques, and also because Missourians versus South Carolinians, or that were smokers, or than you had there? But it sounds like our success rate was even faster than you had in South Carolina.
Oh no, this far exceeded all expectations, and I'll be fully transparent. I felt that was the biggest risk I was taking, is will I be able to transition and get people in, and it really blew away my wildest dreams. I think it's both. I was hyper vigilant to the fact that we didn't want to fail because of recruitment, so we just did every outlet we possibly could. But the other thing too is that, Missouri and this area in particular, I think is really ripe for tobacco research and tobacco treatment.
Yeah.
So we have a great opportunity to reach a lot of people, not just for research studies, but to get them treatment.
So Mary, because we're out of time, I'm going to take your role as the questioner, but I was going to say Melissa Terpstra, our new partner, said thanks for getting her involved. And then she also asked a question: do you have a focus or success in recruiting across diversity? So how did you handle that here in Missouri to get a diverse pool?
Yeah, so what we've been doing is that we typically, it was better in South Carolina, but what we do is we try to match the census the best we can. But we've I'd say outperformed the census, the local census, in our recruitment. And it's really been outperformed in regard to say racial minorities here. One of the biggest things that I think that we face, and others will face as well, is doing these kind of research projects, is that we have to be open to getting people in from outside of Columbia. Yeah. And if we do that, then we get more diversity.
Yeah. Great. So I think I think that's it, Brett. Thanks so much again for giving this great presentation. When we do this again in a month, we have a visiting lecturer, Dr. Hemichandra Reddy, who's going to talk on healthy aging, lifestyle, and reduced risk of dementia. He's currently in University of Texas Tech Health Science Center in Lubbock, and he's a big Alzheimer's dementia neurodegenerative researcher, among other things. He's a brilliant guy, and we asked him to do the November 16th lecture. So that's it. And for all of you who haven't noticed over the last month, Mary Hindle is now Mary Christie. Congratulations, Mary. So, but I think she still answers to Hindle in her email. So, all right. Thanks, Brett. Thanks so much. Thanks for everyone for listening. It was great.